So it's just so cool to be here. Thank you to the organizers for inviting me, and I'm really excited to share some of my work. Although I must say, I kind of feel bad that we haven't solved everything. We had so many wonderful speakers with so much promising data and new insights, and I'm like, ooh, we're going to have to work harder and see if we can get something more out of these monkeys. Squeeze the monkeys. So I'm going to talk about delayed aging by caloric restriction, and we do work in monkeys and in mice and some work in cells. And I guess the 30-second version of my talk is, it's metabolism, dudes. And so we could all just finish now and you could ask questions, or I could show you some data. So I think I'll do that. So these are the monkeys from a 30-year plus study of aging and caloric restriction. These are the ones from Wisconsin. There was a parallel study at NIA, and I think you heard from Rafa the other day, although he didn't touch on the monkey work. So this was set up even before I joined faculty. Despite my gray hairs, I wasn't there at the start of this study. And we were able to show some years ago, and then, finessing or, updating the data as more animals died, a significant impact of a reduced calorie diet on age-related survival and all-cause survival. So this was adult onset. The animals were full stature at the time of coming onto the study. And while it was initially 30% CR, that's not what was maintained the whole way out. There's a natural decline in food intake as animals get older. But we had significant differences in survival and morbidity, age of onset of first age-related disease. So very effective intervention in that regard in our hands. We were really lucky because we had a bunch of measures that were taken over time. So I'm just showing you I had this 3 different groups of animals that were introduced in a bit of a staggered fashion. And I'm showing you controls and restrict there. We had biopsies from a subset that were at a very narrow time window. So that's allowing us to look at caloric restriction without aging being a component. And then we had a necropsy, we collected the whole harvest. So all the rhesus pieces are in my freezers now, including the brains, and that's like really exciting, so we can start to try and piece these things together. We had body weight, body composition, glucose, insulin, blood chemistry, inflammatory panels, and a whole bunch of things that we were able to measure as time went through, more clinical and medically focused things, but we retained all the specimens from those longitudinal captures, so we can now do, for each animal over the course of its lifetime, plasma lipidomics, metabolomics, proteomics, and we have those data now, but I'm not going to show them to you because it's a hot mess. I didn't say that out loud. Monkeys are heterogeneous, and then not only that at their outset where the data are all over, they also age, so that they get all over with age and all over because they are genetically individuals and not inbred. So it becomes a very interesting challenge to pull the biology out of what could be called noise, but I don't think it is. I think the biology is hiding in there. So I've got increasing numbers of mathematicians and computer scientists and engineers in the lab because we're trying to wrangle these data. So we're in a great spot though, and I can show you these from our study. These are all different diseases that were either delayed in onset or prevented in the caloric restricted animals. As these animals, this is the exact same food, they're just eating less of it, okay? So we're not doing anything to do with diet composition, it's literally energy intake. The nice thing as well, and a couple of people have mentioned the Caloric study, which I just really love. This is the human trial of caloric restriction over 2 years in healthy individuals at 3 different locations across the US. I think they're doing another one now, which is going to be really cool. But what you see with human CR is just what you would see if you were a mouse. And as it turns out, if you were a monkey. So lower body weight, lower adiposity, glucose, insulin sensitivity, all looking jolly. So that's nice in terms of that translational component. The overt features of CR are retained between mice and monkeys. So one of the things we became interested in is adiposity. And the idea, it's not really the fat mass is what's counting, it's fat function. And this explains the so-called obesity paradox, which isn't a paradox at all if you ever opened a book. It's this idea that getting away from this size and scale of the adipose and asking, what is it doing? So we became became interested not just in the adipose tissue response to CR, but if there was depot specificity in that response. So we all know visceral fat bad, subcutaneous fat good. It's a little bit simplistic, obviously, but we wanted to know if they were doing the same thing. Well, it turns out that these two depots are quite different. They use the same transcriptional toolkit, but the rank order is not quite the same. And one of the striking things, so V is visceral, S is subcutaneous. When you look at the differences that separate them, some of these chromatin remodeling factors like polyclone and Hox genes are employed differently in the two depots, and we've no idea why that is. So they establish their adiposeness in a slightly different way. There are particular pathways that are enriched in subcutaneous and not in visceral, and I'm showing you there. Metabolism primarily, and it's true that you do see more inflammatory signatures from the visceral tissues. They don't respond the same way to caloric restriction. There are shared features. In particular, I'm loving the spliceosome these days. It's really, really interesting. We see RNA processing as a thing popping out with age, but specifically and used in a different way with caloric restriction. Proteasome and proteostasis always show up in shared. And then there's this intriguing ribosome business that we see all the time with aging, with CR. And we don't fully understand what it is. So we can't say ribosomes up, ribosomes down, but we probably think ribosomes different. And I'll show you a little bit more about that. Those pathways I'm showing there are the two different the responses to CR, and you can see quite different depending on whether they're subcutaneous or visceral. We have RNA processing evidence for it. It's more evident in the subcutaneous depots than it is in the visceral. In addition, when we go in and do exon counting, so this is, exon skipping, exon counting, and swapping and transcript processing, that we see specifically in the subcutaneous. And interestingly enough, it's completely restricted to transcripts that are involved in growth and structure. So it's very fascinating that the mechanism of modulating how that processing is going on is somehow constrained to those cytoskeleton and ECM receptor interaction pathways. How is that happening? We don't know. And then I showed you, mentioned already, alluded to that ribosomal consequence, and it's really very striking. We don't know the biological significance of that. Is it a change in composition? Does it change which transcripts get there, how they get to stick around, how many proteins are made from each one? We just don't know. But it's absolutely fascinating. Monkey and human adipose are very, very similar. So we did a quick run. I say we, I mean my scientist in my lab, Joe Clark, he's so fastidious. I have to try and get him, stop downloading more data sets. We're done. So we compare for studies that had subcutaneous and visceral together. And I'm showing you there in this nice rainbow-colored, you can see here the one on your left. You can see the very nice alignment with what we see in our monkeys, which are the red circles, and what we find in human studies. Where they diverge is when the two depots were taken from morbidly obese individuals. But for healthy individuals, nicely aligning what defines subcutaneous at the transcription level versus visceral. We were also happy to get the adipose data from the Caloric studies. These are the people in CR. And we can see there now they have just a short-term response, and the data I'm showing you here are actually necropsies, so they're old animals. But you can see, apart from that middle section, very good alignment in what we're seeing in subcutaneous adipose tissue response to CR. So it's very conserved. And this, I just couldn't believe this. I thought Joe made it up. Look at the alignment for the ribosomal pathways. So at one point I was like, this can't be, we're seeing it everywhere. We see it in mice, we see it in humans, we see it in monkeys. I see it when I close my eyes. So I just close that bit off there. So adipose tissue definitely responding to CR. Good set of translational conservation between humans and monkeys. So metabolism had been on the radar, and I'm interested in adipose because it's an endocrine organ. It secretes factors that regulate metabolism systemically. It's part of that communication network, not just a bag of fat. And so this metabolism theme has emerged across all of the tissues that we've looked at. And it was very little, you can see this is published already. This was a short-term CR, only 2 years in, from a subgroup of our monkeys, and we had these liver biopsies, and we were able to do a whole bunch of molecular profile screens. The interesting thing is that when you see that, there's a clear pattern that is induced by caloric restriction. It changes metabolism in the liver. And parts of the program are distributed across transcriptome and proteome, lipidome, and metabolome. So when you regress all of those molecules together and some post-translational modifications, they form these clusters, these mega clusters. But the key thing here is that the molecules of different sorts are within each cluster. So it's not one mechanism to induce this response. It relies on transcriptome and proteome and RNA processing, and it's evidenced in lipids and soluble metabolites. So very coordinated and distributed among the things that we measure separately, but you have to put them together to see the story. And I show you the network there, and you can see these are the key features. All the little circles wearing blue eyeliner there, you can see the blue circles, are subject to RNA processing. So the central nodal network introduced by CR in liver is heavily influenced by this RNA processing phenotype, and it distributed all across metabolism. Immune and inflammation generally down, metabolism altered. So I want to avoid saying metabolism up, metabolism down, because that doesn't make any sense at all. It's metabolism altered. In skeletal muscle, we also see preservation of muscle mass and function with age in the monkeys. We had actigraphy measures, we had metabolic chambers, double-labeled water, and then we could also do DEXA imaging for estimated muscle mass. They retain their muscle mass, so the animals are smaller, they're smaller muscles, but they retain the mass peak for longer, and then the pace of decline is more gradual. And so we were able to look in and see, and every time we looked in, we could see connections to whole body metabolism. So I'm showing you a couple of plots there. I mean, it doesn't matter. You can look at it if you want. I mean, go to the paper. We see a connection between preservation of muscle mass with the metabolic cost of movement. We also see preservation of muscle mass connecting with insulin sensitivity. So peripheral indices of metabolism connect to that preservation of muscle function. And we can put a whole bunch of things together, and you can see in that lower quadrant in the multiple factor that we call that Tim's pizza, because he makes these for all of our data sets and they always look like pizzas. But this is multiple factor analysis, the circle here, and you can see insulin sensitivity connecting with that preservation of skeletal muscle mass. I'm only going to touch on this because it was Jen's talk the other day, and I just really want to bring up this: females are not like males. When you look under the hood at the molecular level, we're seeing very distinct patterns of age and also the response to CR. There is a common theme, it's metabolism, growth and repair, but the details of how aging occurs, even though manifestly at the physiological level aging is aging, under the hood it's not the same. So it's really important that we delve into this sex dimorphism. I think it's going to be very revealing and clinically super relevant. So we also had brain imaging done. I'm just looking at my time here. And so this was really fun. We were able to identify preservation of gray matter volume in these monkeys by MRI. We could go in, and what was interesting was that preservation of gray matter volume connected, in some regions but not all, with peripheral insulin sensitivity. So the periphery is part of that regulatory process in brain aging, so metabolism. White matter tracks, we were also able to show greater integrity in the CR animals, but now the connection was with circulating lipids and adiponectin, which is an adipose-derived hormone that regulates metabolism in target tissues. We were also able to look at brain glucose uptake, and here again, the preservation of glucose uptake, and I think this is different from what Linda was mentioning earlier with the inflamed and disastrous high glucose. This is just a gradual decline with age that we see in monkeys, and that preservation is also linked to peripheral insulin sensitivity. We can look at the transcriptomes from the cortical region of these animals, and we see a nice metabolic signature. And we can see, I love this brain in a blender. If anyone doesn't work in brain, you really should, because then you can use these cool tools that have been generated and given freely for you to go in and take a look. So you can see there all the individual animals and the contribution of different cell types. So we're following this up. We see the biggest changes. There's a good variance as a function of age. Neurons seem to be preferentially responsive. So we're interested in different cell types and how they connect. The proteome tells a similar story, but not the same story. So again, this idea that different molecular profiles will give you, it's like a different view into the same room, okay? So we see pathways, of course, metabolism all over the place, and we see the spliceosome again showing up, and we see nice accord there as a function of age in those age-sensitive modules, and this was WGCNA. Thank you, Steve. I told you that we use this all the time. Cautionary tale, I'm not going to say much about it, but someone wants to ask later. You can't assume anything from one ohm to another. The transcriptome will not tell you what's going on in the proteome. And in some cases, and this is, Joe being fastidious, they're completely opposite in terms of increased or decreased abundance or level of enrichment. Not all of it, but a lot of it. So one doesn't inform of the other, you need to do both. So I just need to do a very quickie whirlwind. So my interest is in metabolism and how do we get it. And I love what Brian was saying earlier about we really need the basic biology to come in. We've got this physiology, we've got these clinical indices, we have even effective treatments, but how are they working? So this is like super fast, even faster. It's energy restriction, okay? So this idea that energy intake is related to regulation of longevity, at least in the context of caloric restriction, which means things that respond to energy, detect energy, distribute energy, store energy, are going to be responsive, and they're part of that longevity regulation. We did a quickie experiment, and this idea was, look, okay, if mitochondrial function and metabolism is all that, why don't we just mess with it a little bit and see what happens? And what happens is, you can take these cells, this is PGC1 alpha, I think it was mentioned earlier this morning, to dial the mitochondria up a little bit. I'm not going to show you the data, but we were able to do that and then show changes in whole cell redox, RNA processing, chromatin remodeling, cell size, cell proliferation, cytoskeletal dynamics, growth signaling, and lipid handling. And all we wanted to do was dial the mitochondria up a little bit. The point is, when you change metabolism, everything else responds. And this is why I think that metabolic component to what's happening with CR extends beyond those initial just measures of metabolism to influence all kinds of cellular function. And this is how I would sum it up here. These are all the hallmarks currently. I left a few out, don't blame me. And they all have a component of metabolic dysfunction, and our work has shown in the areas where we've looked, you can change metabolism and mitochondrial function and impact these other features. So put it at the center. In mice, we see a similar sort of setup. It's metabolism with age. That's really nice. We see the biggest signature is inflammation with age. The second biggest signature is neuronal function with age. The third biggest signature is metabolism. And it's spread across all different types of metabolic pathways. We see a consistent story, but not identical in the proteome. I'm showing you here. And these are from the exact same specimens, because we can split them and do both profiles from the exact same thing. So they are directly equivalent. But this story is that we have inflammation is happening with age, and metabolism is in a state of, I suppose, adaptation. I wouldn't call it decline. The brain is weird because PGC1 alpha, the big regulator of mitochondria, is differentially expressed depending on the cell type. There are 3 different promoters, and neurons are so extra special. They really think they're all that. They're so extra special, they get their own PGC, run off their own promoter. And that's super interesting because it's regulated differentially than the others. But here's the thing. We've been looking at this, and we can see there's the change as a function of age in PGC1 alpha expression, and whether you look at a version from the canonical or the alternate or the brain-specific promoter, that decline is happening, potentially explaining what we saw in the brains with age. So we thought, okay, can we do something with PGC and actually impact something that is pathological, right? So we had this Alzheimer mice. These are the tau mutant mice that they get this pathology at about 6 months of age. We had those neurons, and we were just tooling around, and we tried, well, what's going on with these neurons? We found that they had very much lower PGC1 alpha levels. And we had a drug that we thought, we knew we could target PGC via AMPK, so we thought, okay, let's do this. Let's dial those mitochondria up a little bit. We were floored, because we did dial the mitochondria up a little bit, but we also removed the phospho-tau and cleared the neurofibrillary tangles. So what the heck is this going on? So we dug in a little deeper, and it turns out by targeting that PGC axis, not only did we change metabolism, I'm showing you here in that heat map of the transcriptional profiles, there's metabolism all over, but we also see things like ribosome coming up, DNA replication, base excision repair, and then these other aspects of immune function. These are just neurons by themselves. So we're changing diverse features across the neural profile. We see changes that we would have predicted, membrane potential, ATP synthesis. Tau neurons have these bizarre, huge lipid droplets, and we don't know why, but we can target PGC1 alpha with this drug and alleviate, we can reverse that problem. It turns out that metabolic deficiency or disposition that's different extends to other aspects, cell morphology, which is tau in these tau neurons, and we can repair that also by targeting this PGC axis. You can see it there. And then another unexpected finding was that we were able to patch clamp these neurons, and we could show deficits in their responsivity because of the tau burden, and that was also alleviated when we target this CR axis. So the upshot is CR induces changes in metabolism, cellular level, and whole animal levels, and we see it in all the signatures that we look at from mice or monkeys. But can aging metabolism explain age-related disease? And I think the answer is yes. And so this is a study from a couple of years ago. This is a perspective piece that Tim and I put together describing that study, and it shows how metabolism, metabolic rate changes over age for human beings. And so everything's stable between 20 and 60 years, but then things start to go into decline. And that's around the same time where you see that decline in adiposity. And I don't mean, you know, boy, don't I look great, I've lost weight. I mean that I don't want to lose weight, lost weight sort of thing. And those are coincident then with the increase in disease vulnerability as a function of age. So we think that all of these things are connected. It's metabolism, baby. Right? So I want to thank my lab team. They're fantastic, former members, current members, collaborators, and my funders, absolutely super. They're the kids on the block. And I'll take any questions, if there's time.
Thanks for a very fascinating talk. Maybe we have time for a very quick question.
Thanks. I have minimum 10 questions, but just one. So the ribosome story is very cool. Do you see differences between the mitochondrial and cytoplasmic ribosomes?
We haven't dug into that. So in the monkeys, you know, the tissues are frozen. It's going to be very hard to get organelle-specific components, but we can try and recapture some of that in our cell culture models. But right now, we're at a loss. We just see it everywhere, and I don't know what it means. I have to partner with some ribosome and RNA people. I think it's about time, yeah. But thanks.